For the second section of this chapter, begins discussing microscopes, how they function, how they work. I'm not going to spend a lot of time on this. Actually, I'm going to spend very little time on this. I invite you to read those pages. What I will do is point out a few things. Point out the reason we need microscopes. If you look over here, the arthropods, the lice, the sporangiosome of fungi. While we can just make them out with our naked eye, you might need a magnifying glass to actually see them better. But when you start looking at much smaller things, things that are a single cell, like Giardia lamblia here, or a red blood cell. Now we're in the point where you need a light microscope. A light microscope is going to use the light from a light bulb, pass it through the specimen on a glass slide, through objective lenses, through the ocular lenses, and then you see something. As you go up in magnification, though, you're going to be losing resolution. So at low magnification, the Giardia, the red blood cell, will look nice and crisp. But as you go up magnification so you can start to see, things within the cell, you're going to lose resolution. It means it's going to be less crisp as you look at it. So you can continue up magnification to about 1,000 times so you can see individual yeast cells, individual E. coli cells. But the thing is the resolution. To be able to differentiate between parts is going to get harder and harder. At that point, you're going to need something like an electron microscope. Electron microscopes come in two forms: transmitting and scanning. Scanning bounces a laser. Forget light bulbs, it uses a laser beam. Bounces a laser beam off the surface of Giardia, red blood cell, or E. coli to see what the surface looks like. Gives you a nice three-dimensional picture. The transmitting passes the laser through the cell. Anything in the cell, organelles, membranes, proteins, ribosomes, will cast a shadow. So you're actually seeing the shadow of the cell, seeing the shadow of the things inside of it. The transmitting electron microscopes allow you to see things like individual viruses, protein complexes. We're soon going to be at the point where we can start actually seeing DNA. With these microscopes, these electron microscopes, magnification can get very high, tens of thousands to millions, and the resolution will stay nice and crisp. But say you want to look at something. Say you want to use the light microscope, and you want to look at living cells. You want to look at protozoa. You want to look at a sample from a pond. You want to see what's there. Well, you would use what's called a wet mount. A thick glass slide that has a depression ground into it. You take a cover slip, you put a drop of your pond water. You turn it upside down so that the drop just hangs right there in the depression. And you're going to be looking from the top down. You're going to be looking in that general direction. What you're going to find is that water droplet's just going to hang there in all the cells, all the protozoa, all the fungi, whatever, are going to be free to move, free to swim. So you can see locomotion, how Tetrahymena swim. But that's only good for eukaryotic cells because they're much larger. Bacterial cells, which are about 100 times smaller, well, they're so small, light passes through them, light bends around them. So they're almost invisible in a microscope. So what you have to do is fix and stain the bacteria. Robert Koch, Koch's postulates, Robert Koch, came up with this means of smearing bacteria. You use a loop, you use something, you pick one of those colonies from the last lecture. You smear it on a glass slide with a little bit of water, smear it out. You let it air dry. And then you heat it over a flame, a Bunsen burner. What this does is it drives off the water. Once it's dry and you're heat fixing it using the Bunsen burner, you're literally melting the bacteria. The sugars on the surface of the bacteria, you're melting them to the glass that allow you to stain the bacteria. What was invisible will now be visible because of stains. Stains can be added to these smears to colorize the different cells. Or on the flip side, to colorize everything but the cell, so that you can see what the cell has around it. Does it have a capsule? Does it have a slime layer? Dyes impart color to the bacteria. Different dyes, different colors. The different dyes not only have different colors, but they have different chemical properties. The different chemical properties will allow you to do either negative or positive staining. You look at the table on the right there. Positive staining means you're going to stain the cell. As you see down here at the bottom, these little blue rods are bacterial cells that have been stained with crystal violet. Crystal violet collects in the cells, and then you rinse off everything that's not in the cell. There you go. You can now see the shape of the bacteria. There are multiple kinds of these positive stains: crystal violet, methylene blue, safranin, malachite green. These positive stains can be simple or differential, or as you see at the bottom there at that column, special stains for finding out whether or not there's flagella. Can it form a spore? Can you just stain the nucleic acid? You counter this with the negative stains. As you see here, everything around this bacteria, that bacteria, the little dots here, which are little small bacteria, everything but the cells are stained. Nigrosin, India ink will allow you to tell whether or not the bacteria has a capsule, a protective coating around it. A protective coating that wouldn't show up in a positive stain. Now, the positive stain comes in two forms: simple, differential. Simple, it's a single dye, as you see there on the right. Crystal violet, staining E. coli. The different rods are E. coli. The little stuff you see around it, that's just the glass holding on, dirty microscope slide. But you can see the shape of individual E. coli bacteria. You look over on the right there, methylene blue, staining Saccharomyces, yeast. You can see the shape of the individual yeast cells. Heck, you can see up here, some of them are reproducing being budding. You can see the shape, but that's about it. Differential gives you different abilities. So, simple stains, single dye, tells you just basically the shape. Differential, well, just like differential agar lets you tell the differences between bacterial species based on color of the colonies and things like that. Differential here for stains allows you to tell differences between bacterial species based on what color the bacteria will be. It uses a primary stain and counter stains. You look right here. Red rods, purple spheres. This is the Gram stain based on thickness of the cell wall. They're either going to stain blue or they're going to stain red. Here in the middle, this is the acid-fast stain. Cell wall here is going to be made of wax. Does the bacterial cell have a waxy cell wall? Yes or no? This is basically used for, staining Mycobacterium tuberculosis, the bacteria that causes TB. When you look at the Gram stain was developed by a gentleman by the name of Hans Christian Gram. Yeah, you want to be immortal? Discover something. Name it after yourself. What you see here is Gram developed this system of primary stains, decolorizer, counter stains, that's all based on whether or not a bacteria has a thick peptidoglycan cell wall. If it's a thick cell wall, it's Gram positive. It'll be purple. If it's a thin cell wall, Gram negative, stains red. Based on that and an idea like that, like I said, the acid-fast testing for mycolic acid. Forget peptidoglycan, we're looking at a wax. You're going to find this in Mycobacterium and Nocardia species.